Okay, this lecture is going to take you through the outcomes for microscopy, or looking at cells with a microscope. So we're going to focus on two different types of microscopes: the light microscope and the electron microscope. So both are used to magnify or increase the size of images so that we can see things that we normally cannot see with the naked eye. Light microscopes are the types that you use in your laboratory classes. You've probably used before. They use light as the source. They're also called bright field. And you've probably seen a microscope that looks something like this. You have objectives that you can change the level of magnification, and you have eyepieces that you look through. Electron microscopes, on the other hand, use electrons to create the image, and so you can magnify things even more. You most likely have not used an electron microscope. It's this big looking machinery, costs hundreds of thousands of dollars, and you have to treat the samples with heavy metals in order to visualize them. So big difference is, and you're going to see examples of this, the electron microscope, besides using electrons to create the image and getting greater magnification, you get much more detail of the sample. Okay. So these are actually pollen grains. Pretty cool looking. And this is and I always spell it wrong, I think. Amoeba. Maybe I spell it right. Okay, so let's look. So we're not going to go into all the details of how these work. Your objective for microscopy is to describe the differences between light microscopy and electron microscopy. Think about how each works to obtain an image. So one is using light and one is using electrons. And what types of cells, cell structure can you see with each? That's what we're going to go through next. Your second objective with microscopy is to describe and be able to recognize the different types of images you get from a TEM or SEM. So TEM, SEM are types of electron microscopy, and we'll talk about those. And then you're also, you want to be able to define magnification and resolution. So we're going to go through all of those. Okay, so what can we see with a light microscope? Okay. Well, one thing we can look at is we can see both prokaryotic and eukaryotic cells. And we can see some, very few, organelles. So let's take a look at the image on the left. This is an image of a cheek cell. And the dark spot you see here is the nucleus. So this is an example of an organelle you can see with the light microscope. This is the outline of the cheek cell. So there are two here. Here's the other nucleus. And then all these little dots are bacteria. So this is all the bacteria that's in your mouth. So the way we obtain a cheek cell is you can take a toothpick, scrape the inside of your cheek a little bit, put it on a cell, and then you're going to stain it. So in this case, these cells are stained, so normally your cheek cells are not blue, and since the cells are stained, they are dead. Okay. So this is an example of where you can see both prokaryotic and eukaryotic cells, and you can see the nucleus as an example of an organelle. One thing I want you to see is look how small bacteria are compared to your large cheek cell. Okay, you can also look at live cells. So on the example on the right, this is a plant cell. I'm outlining a single plant cell. Here's another one. They kind of look like bricks. So this is plant. And these green little globs, those are chloroplast. And what's really cool, and I'm not sure if you guys get to do this in lab or not, but you can put plant cells under the light microscope, and as the light starts activating the chloroplasts, they'll start moving around. It's called plasmic streaming, and the chloroplasts are trying to find the best location to gather the most light for photosynthesis. So that's pretty cool. We can see these organelles because they are green because of the chlorophyll in the chloroplast. The bottom picture here, this guy is a paramecium. Okay, I don't know why I say he. It is not an animal cell like your cheek cell, not a plant cell, but a protist. This is another group in the eukaryotics. It's another family? Kingdom? I don't remember all my taxonomy. Here you can see some distinct shapes, right? But it's really hard to tell what they are. Some of these are probably vacuoles that are storing. And if you look really carefully, you can kind of see this fuzziness, especially at the ends, you can see it. These are the cilia. So remember, cilia are short protein structures projecting. And you can kind of see them here. I don't think I drew the cell very well. Projecting on the outside of a cell, excuse me, and the paramecium use their cilia to move the environment around so that they can eat. Okay. So, again, with light microscopy, we can see both prokaryotic and eukaryotic cells. Prokaryotes are very, very small. We cannot see anything inside that prokaryotic cell. You can look at live cells, or you can look at dead cells that you stain. And again, this paramecium is live. It's not stained. Sorry. And so it makes it hard to distinguish the different parts of it. So you can see some, but very few organelles you can actually make out. An electron microscope, remember I told you you can see much more details. In fact, the magnification level for electron microscopy is about 100 times that of light microscopy. These guys in the middle here, these are viruses. You cannot see a virus with a light microscope. If you want to know, this is a phage. This is a type of virus that infects bacteria. And this little guy is influenza, the virus that causes the flu. Okay. So that's one big difference. You can see much greater magnification, and you can see much greater details. So in fact, here is a picture of a mitochondria. Oops, but I can't spell it. All right. So mitochondria, remember we drew, I'm going to change the color here so we can see it better, as an outer membrane, and then we draw it like this, right? But what that is hopefully you can kind of see all of these invaginations, or these little folds, these cristae of the second inner membrane. So that's a mitochondria. You can definitely not see the details of mitochondria with a light microscope. This here is a cell, so let me outline. So that is the membrane of the cell. This big guy is the nucleus. This area in here is the nucleolus. We've got some vacuoles that are storing things. If we zoomed in, you probably, some of these are mitochondria, ER, but if you compare this, whoops, to this image, right, you have a lot more detail. Okay. These two, on the left-hand side, is what we call transmission electron microscopy. So these are TEM images. And transmission electron microscopy allows you to see kind of a cross section. I didn't leave myself much room to write here. Of the cell. Because remember, cells are 3D, right? Three-dimensional. So you're looking at a cross section cutting through the cell. In contrast, on the right-hand side, this is called scanning electron microscopy, or SEM. Scanning is giving you the 3D image, which is the surface of whatever you're looking at. So again, these little spiky balls are pollen coming off or attaching to a plant. This SEM, these two guys, are bacteria, right? So look at the detail. You can see these are bacillus, means rod-shaped bacteria. Look at that compared to the highest magnification you can get with light microscopy, right? So lots more details, and you can see it looking like 3D. You're getting a surface image. Okay? Transmission, you're seeing the inside and you're getting a splice. Scanning, you're getting the outside and 3D. So big differences between electron and light microscopy. Electron, you see much more details. You can see surfaces. You can see the inside of a cell, and super cool, you can see viruses. All right, so I just wanted to show you, well, what do cells really look like? So these are prokaryotic cells. So again, here is a cheek cell, and you've got the nucleus right there, and all of these little dots are bacteria. Okay. This is light microscopy. This is light microscopy. This is light microscopy. So these are bacillus, little rod-shaped. These are circular cocci. I can't tell if this is staphylococci. It looks like So like a staph infection. This one up here, this is electron microscopy. This is scanning electron microscopy with false coloring. So schools it would be staph, Staphylococcus, like epidermidis or whatever this guy is, not purple. And you noticed up here all of the electron microscopy I showed you was black and white images. We can add color to give it more contrast, and so you can have false coloring. So if you see great detail, and this is a scanning because you can see the outside the 3D, that is scanning electron microscopy, SEM, with false coloring. Down here we have our classic black and white, right? So this could be, I'm guessing this might be light because of the glowing around it. These are Vibrio, these are prokaryotic cells, and here is for sure an electron microscopy. I would guess this one is scanning. That looks really silly. Because you can see kind of the 3D shadow. Here are some flagella. This little guy might be Helicobacter pylori, might be Vibrio, but these are prokaryotic cells. They are bent like this. You can see how much bigger they look than with the light microscopy, even though this is cocci. These are things that you could see images of prokaryotes under one of the different types of microscopes. Okay, what about animal cells? So I always think it's funny, but we just do it traditionally. We draw an animal cell as a circle, and then we put all the parts in it. I want you to know that all animal cells are not circular. So this is a neuron, and you can see the nucleus here, and then all these long projections that come off this neuron for cell communication. These are red blood cells, and the monocyte in the middle is a white blood cell. White blood cells are really big, and they have this weird shaped nucleus. This is one of the ways you identify them. Red blood cells actually have lost their nucleus. So they look like these little pillows, little kind of donuts. They are a whole cell, just kind of how the light is reflecting. So these images are all taken with light microscopy. Here is epithelial cell, so your skin cell. So this could be one of those cheek cells that I showed you earlier. The nucleus, you can kind of see this dark spot, that's the nucleolus. Over here, these are cells, should have had my notes. These are either cells that line your, I think these are lining your trachea, that have the little cilia coming off of them. So that helps sweep out things that you breathe in. You can see this big nucleus, and this is the outline of that specific cell. And then below it you can see other cells. This here is muscle. Long fibers, and the purple stained is the nucleus. So animal cells come in all shapes, different sizes, and again with the light microscope, really the only detail you can see is the nucleus, and you may see a dark spot representing the nucleolus. What about plant cells? Again, plant cells. We always draw them like this, and we put in all the organelles, right? Okay, so plant cells come in massive varieties. So here's a similar picture to the one we looked, and they look a little less brick-like. Right, these are cells in a leaf. All the green things are chloroplasts. This is cross-section of a root and a stem, and they're false colored. But I want you to see how all these cells are packed together. Due to the staining, they've lost their internal organs. These cells here are on the underside of a leaf. This guy, who I think looks like lips, let's try to draw that again. So there's actually two cells. That's the stomata. I love these guys. They open and close to let oxygen out, sometimes water out, and CO2 in for photosynthesis. So we'll talk about stomata. This one is my favorite. Looks like a bunch of jigsaw pieces. These are also plant cells. You can see the bright green is the nucleus, the outline of the plasma membrane. This was an image that I first saw when I was a postdoc, right? So I had gotten my bachelor's degree, gotten my PhD, didn't really ever learn a lot about plants, but switched over to studying plant viruses. And one of the images they showed me when they were showing me the research is a picture like this, and I was like, oh, that's so cool, what is that? And they were like, plant cells. And luckily I kept my mouth quiet because I thought plant cells were squares. Not good, impressing your your postdoc advisor, but quickly learned that plant cells are not these goofy little squares like we draw them. They come in all different shapes and sizes. Okay, so now you've gone through and you've listened about the differences between light and electron microscopy. You've looked at the different kinds of images we can see with the two types of microscopes. The last information is I want you to make sure you understand the difference between magnification and resolution. Okay. So magnification is the size of the image. Right? So how zoomed in are you? Okay. And you have changed magnification if you've worked with a microscope by using different objectives. And you might have used a 4x and a 10x and a 40x, and if you were lucky you got to use the 100x objective, which means it was magnifying things 100 times, 40 times, 10 times. Also in a light microscope, the ocular that you look through, let's see if I can draw an eyeball. Oh, no, you're going to have eyelashes coming out of your eyeball. That would be kind of gross, right? Eyeball. The ocular usually has a 10x magnification as well. So with a light microscope, you usually can go 10 times 100, 1,000-fold of magnification, whereas with an electron microscope, you can go 100 times more. Right? So you could go 100,000 times of magnification. Okay. Magnification is important when you're comparing cells or organelles or different sizes of things. So you've got to know what magnification the image was taken at. And these I have not given you the magnification. So this is actually very low magnification because you can see so many different cells. This is very high magnification because you're seeing just a few cells, and the cells are huge. So going down the picture of the dog here, you get different levels of magnification. You also have different levels of resolution. And the definition of resolution is the ability to see two items as separate discrete units. Right? So this has higher resolution than this image that's blurry. This image has higher resolution. You can see all of these bumps on the dog's nose where, all right, high and low resolution, where it's very blurry. And resolution is basically based on how good of a microscope. So microscopes we use in class don't have as great of resolution as microscopes they may use in a research lab. Just costs more. Okay, so that covers your microscopy outcomes. Next, I want you to kind of get a feel, and I want you to test yourself on what kind of images. So one of your objectives is be able to recognize the different types of images you get from light and TEM and SEM microscopy. Okay, so what I'm going to do, or what I want you to do, is look at these images, maybe pause, decide what your answer is, and then listen to the answer. So based on the following information, what type of cell is shown in the image? And hopefully you chose eukaryotic cell, because if you see nucleus, you know it's a eukaryotic cell. So I didn't ask you if it was plant, animal, protist, but it's definitely cannot be prokaryote because there's a nucleus. So it couldn't be either type, and because there's a nucleus, it is eukaryotic. Okay, what about this one? Green color is due to chloroplasts. Chloroplasts are an organelle. I don't know why this pen does this. Right? Therefore, it is also eukaryotic cell. Okay? Prokaryotes can be green, but they don't have chloroplasts. So it can't be either, and it is eukaryote. This is my favorite little guy, Euglena. This is actually a protist. So he has it has chlorophyll. What you can't see is it also has flagella that allows it to swim. They're really cool little guys. Okay, take a minute and see what you think this is. Okay, so it says we have an envelope, we have a lipid envelope, tegument, DNA, nucleocapsid. It hasn't told us anything about organelles. It hasn't told us anything about a plasma membrane or cytosol. This is in fact a virus. This is neither type. A virus is not a cell. A virus is basically a protein and DNA or RNA genetic information, but not a cell. So this one would be hard. You wouldn't see one like this on the test, but I like to challenge you in class. Okay, based on the following information, what type of cell is shown in this image? Okay, this could be either. It has ribosomes, it has a plasma membrane. Both eukaryotic and prokaryotic cells have ribosomes and plasma membranes. So based on what I've told you, this could be either. This is in fact a prokaryotic cell. There's no nucleus, but you don't know that. So when you're answering questions, you have to answer it based on what I've told you, given the information I've given you. What about this guy? Take a look. Okay, so this has cells and it's green, but we really don't have enough information. So you would have to answer could be either. And just so you know, this is actually a prokaryote. This is cyanobacteria. So this is some of these cells are examples of photosynthesizing bacteria. But based on what I gave you as far as information, you couldn't tell. What type of microscopy was used to generate this image? So you're seeing this cool 3D looking kind of monster, right? This is made from a scanning because you can see the outside of this. I think he's a little eyelash mite. Yeah, can you imagine now? Those are crawling in your eyelashes. That guy's made. This image was taken with a scanning electron microscope. Okay, so way more details than you could see with light, and because it's 3D, it's not transmission. And really good microscopy is actually not a valid answer. I like to throw kind of fun things in there once in a while. What type of microscopy was used to generate this image? This is generated with a light microscope. Again, colorful is not an actual type of microscopy. These are white blood cells with that funky nucleus and bacterial cells. Because you can't see a ton of detail, light microscopy is the correct choice. What type of microscopy was used to generate this? This is actually a type of light microscopy, so you're not seeing the details that you could see with electron microscopy. This is actually a high-powered one called confocal microscopy, where it's using antibodies to stain things different colors. Again, this is a neural cell with the axon and dendrites projecting out of it. Okay. If you had said none of the above, and sometimes I do this in class, and so I would never do this on a test, but none of the above I would also accept because it definitely doesn't look like light microscopy. So it's a very special kind of confocal. And one of the things you'll notice in class is sometimes I put much harder questions on clickers than you would have normally on an exam because I want us to have some discussion. Okay, the last one for you to analyze. This image was taken with transmission electron microscopy. Right? So transmission, because it's 2D, it's like a slice through the cell. It's showing you that there's a Golgi, there's a lysosome, much more detail than you could see with light microscopy. Black and white, again, is not a real type of microscopy. So I hope that has helped you learn a little bit about microscopy, what kind of images you could see, what cells look like under the microscope.